Good morning, everyone. I'm not the real Bjarne. Unfortunately, Bjarne couldn't make it, and I was volunteered to give his talk. So Bjarne gave me the ambitious title of the identification of semiochemicals associated with resistance of Atlantic salmon to salmon lice, and assessment of their effects on lice. And actually what I'm going to present is some very preliminary data that we received last week from part of this project, a preliminary experiment that we did. So, as you probably know, the ability to resist sea lice is a heritable trait in Atlantic salmon, but it's challenging to assess the relative resistance to lice for individuals in the population without challenging them to the lice. And there are these compounds, semiochemicals, that are released from the skin that are associated with genetic variation for resistance. But the mechanisms triggering the release of these compounds and the genetic basis for their action is unknown. So the aim of this particular part of the project is the broad aim of the project is to identify candidate semiochemicals associated with Atlantic salmon resistance. So for this talk, we did a pilot study and we took 400 Bencmark Stoffenfisker fry and they were from 40 families. So they were start fed in 2020. And when they were transferred to Seawater, we sent them to Vesøviken in Norway for challenge testing. And they were distributed across 2 tanks. And we sampled mucus at 2 time points before we infected the animals with Salmonus. And then we counted the lice on the animals at 2 time points post-infection. And we sampled mucus at 2 time points post-infection. So we sampled 2 weeks before infection, 1 week before infection, and around 10 to 11 days after infection, and 18 days after infection. And then from the counts of lice, we identified some animals that were extreme in terms of lice counts. So we had 7 high lice count individuals and 7 low lice count individuals. And for those individuals, we went back to this time point 2 weeks before infection, and we sent the mucus samples from that time point for those individuals to Rothamsted Research for analysis of semiochemicals. We also have another experiment where we're looking at conditioned seawater, and I'll explain that as well, and we're intending to compare the results from this experiment to this other. So for the conditioned seawater experiment, we had fish that were movie families, and these fish were tagged and sampled before they were transferred to seawater. And then we split the fish. The fish we chose because their parents had either a high estimated breeding value for lice resistance, or they had a low estimated breeding value for lice resistance. So we split the fish into 3 tanks for the high EBV for lice resistance, and 3 tanks for the low EBV for lice resistance. And we had 20 fish per tank, and then we sampled conditioned seawater from those tanks at 3 time points. And this will also be used to search for semiochemicals, so compounds that differ between the high and the low EBV groups. So coming back now to the mucus samples that we took, we used a soft silicon spatula and we collected into nunk tubes. We had around 100 to 200 microlitres of sample per fish, And it was quite difficult actually to take these samples without stressing the fish too much. So it was quite difficult to obtain the samples. And then the samples were at Rothamsted. They were extracted using solid phase extraction, and we're using gas chromatography mass spectrometry and liquid chromatography mass spectrometry to identify the compounds in these samples. So this just shows the mean trait values at the 4 sampling time points. And you can see that the body weight of the fish at 18 days after infection was around 130 grams. And we have average lice counts at 11 days after infection of around 19 to 20 lice per fish. And most of these lice were at the Calamus 2 stage. And then at 18 days, we have average lice counts of around 16 to 17 lice per fish. And most of these lice were at the pre-adult stage. So, and if we look at these extreme groups that we created, we have here the average total lice count at 11 days. In the low group, we have 3 lice per fish on average. In the high group, we have 54 lice per fish. So a difference of 51 lice on average, or 18-fold difference in the number of lice. And at 18 days, we have 5 lice per fish in the low lice count group. We have 32 lice per fish in the high lice count group. And so an absolute difference of 27 lice per fish on average, and around about a 6-fold difference. So the results last week came in, and unfortunately, so far, we've used the gas chromatography mass spec, and we haven't been able to see any significant differences between the groups. This just shows 2 individual profiles. So this is one low lice count fish, this is one high lice count fish, and so these are the profiles of compounds that come out over time. Just of interest, there's a compound here which is 6-methyl-5-heptane-2-one, and a compound here, isophorone. And these are compounds that we see that have been seen before to really excite lice. So, but unfortunately, there's no significant differences between the 2 groups in the amount of these compounds. But we've got more results yet to come, and also we have the Seawater experiments yet to come as well. So we'll see. Also, we're now undertaking a larger challenge test, and this will be used to test for the genetic basis of these semiochemicals affecting the lice. So for this, we've got large number of movie families, and the animals are about to be challenged this month. So we have 2,500 fish that are going to be challenged with Alcimonis, and we have 18,000 fish that will be challenged with Caligus elongatus, and we'll be sampling mucus at a time point that we decide on from the other experiments, but probably back here somewhere before infection. And then we'll be counting the lice in both experiments, so we can look at whether the genetic basis for resistance to Alcimonis and Caligus is correlated. So that will be quite interesting. And also we can do a genome scan to look for QTLs affecting lice resistance to both types of lice. The other thing that we're going to be doing in this project is we'll on-grow lice from high count fish, and we'll on-grow lice from low count fish, because we think that the environment in the skin is probably quite different in these different fish. And then we'll test, we'll collect egg strings, will test the fecundity and the infectivity of these different groups of lice. So that will be quite interesting as well. So for follow-up experiments, we're going to test the effect of any semiochemicals that we do find on lice behaviour. So that will be done at Matra with the Institute of Marine Research, and also at the University of Gothenburg with our partners there. And we'll be developing more targeted assays for any of the semiochemicals that we do detect, so that we can have a high throughput method for analysing the samples from the larger experiment. We'll be testing the accuracy of these assays as phenotypes for genomic selection to enhance lice resistance, and we'll be trying to identify genomic regions associated with semiochemical profiles that could be incorporated into selection models to improve lice resistance. So we're hoping that this project will provide assays for potential cheromones and allomones, and provide accurate phenotyping methods that we can use to assess lice susceptibility in Atlantic salmon, and also provide better knowledge and a better understanding about the host-parasite interactions and susceptibility. So I'd like to thank all of the partners involved in the project. So our industry partners were Movi, Bencmark, Cargill, and our research partners, the Institute of Marine Research in Norway, the Rosalind Institute at the University of Edinburgh, Rothamsted Research, Bigelow Laboratory of Ocean Science in the US, and I'd like to thank the Research Council of Norway for funding the work. Thank you.